Good evening. My name is Brenda Tyndall. I am the Executive Director of the Harvard Museums of Science and Culture, where our mission is to foster curiosity and a spirit of discovery in visitors of all ages, enhancing public understanding of and appreciation for the natural world, science, and human cultures. This mission in mind, I am delighted to welcome you to tonight's lecture, Evolution and Conservation in the Deep Sea, co-sponsored by the Harvard Museum of Natural History and the Department of Organismic and Evolutionary Biology at Harvard University. Tonight's speaker is Russ Holtzel, Professor of Molecular Ecology at Durham University and the 2020-2021 Sarah and Daniel Hardy Visiting Fellow in Conservation Biology at Harvard University. Professor Holtzel will discuss his research on some of the key environmental drivers and adaptations promoting the evolution of diversity in the deep sea. Of course, you can use the Q&A button at the bottom of your screen to submit questions anytime during the program, and our speaker will address as many as time allows at the end of the presentation. It is now my pleasure to introduce Darren Card, National Science Foundation Postdoctoral Fellow in Biology at Harvard University, who will introduce tonight's featured speaker. Enjoy and thank you.
Thank you. Welcome everyone, and thanks for being here tonight. I am pleased to introduce our speaker, Russ Holtzel, Professor of Molecular Ecology at Durham University. Professor Holtzel is originally from Massachusetts and actually grew up nearby in the Hingham area before he switched coasts entirely and obtained his bachelor's degree from Reed College in Portland, Oregon. Russ then switched continents entirely by going abroad to the United Kingdom, first for a master's degree in evolutionary biology from the University of Sussex and a doctorate in genetics from Cambridge University. After research appointments at Imperial College London and back in the US at the National Cancer Institute, Russ settled at Durham University in the UK, where he has been since. Together with his research team in the Molecular Ecology Group, Professor Holtzel has been a leader in the application of genetic approaches to diverse research questions in evolution, ecology, and conservation biology. More specifically, Russ's research has leveraged modern genomic methods to understand patterns of genetic variation in natural populations in marine ecosystems. And these investigations have improved scientific understanding of how modern environmental change mediates ecological dynamics that can contribute to the overall evolutionary trajectory of natural populations. In our current era of global change, Russ's major research goal of developing more effective conservation strategies is taking on increased importance, especially given his focus on marine ecosystems and generally on large marine mammals like whales, dolphins, and seals, which have been negatively impacted by human activity in recent centuries. Russ's service to the scientific and conservation communities is also impressive, with highlights including membership on the International Whaling Commission Scientific Committee and a role as the founding editor and editor-in-chief of the Journal of Conservation Genetics. And as we've already heard a little bit about, Professor Holtzel has returned to the Boston area this academic year as the Sarah and Daniel Hardy Fellow in Conservation Biology at Harvard University. And as part of his fellowship, Russ is revisiting the important ideas of evolutionary significant units for conservation, and is using both modern and ancient samples of DNA to profile the genetic and evolutionary patterns resulting from extreme population declines and subsequent recovery in elf and seal populations. Tonight, Professor Holtzel will be speaking on evolution and conservation in the deep sea. So please join me in welcoming Professor Russ Holtzel.
Well, thank you, Darren, and thanks everybody who's tuned in tonight. And I especially want to thank Sarah and Daniel Hardy for this brilliant fellowship that they established. So today I'm going to talk about evolution and conservation in the deep sea. I just wanted to acknowledge the people who actually did the work that I'm going to be telling you about today. Anders, Michelle, and George, all postdocs, and Fatih, intern, and many other people as well, some of whom are listed at the bottom of the slide there. So oceans are vast. They're 71% of the Earth's surface, 360 million square kilometers. This is familiar. It is the blue planet, after all, but maybe not so familiar is just how deep the oceans are. 3.7 kilometers on average, up to 11 kilometers at its deepest point in the Marianas Trench. And below about 1,000 meters, it's mostly dark, cold, and there's high hydrostatic pressure. So it's no surprise really that people considered the deep ocean to be basically a desert, depopulative life for a long time. And it wasn't until the late 19th century that people started to dredge stuff up that gave them the idea that maybe there was more there than they thought, including things like whalebone with new species of mollusk attached. But things didn't really change until 1967 with this paper, where the authors Hessler and Sanders employed what was then a brand new piece of kit, this epibenthic sled, which they could slide for long distances across the bottom, and they did so at 1,500 to 3,000 meters. And they found a great deal more diversity than they had thought was there. And then later in the 20th century, it was possible to build remote operated vehicles like the one shown in the upper right, which could take film and photos and collect samples, and manned submersibles like Alvin down on the lower left there. And they recorded a great deal more diversity and cemented the idea that yes, there is a lot of diversity in the deep sea after all. In the late 1980s, Alvin filmed a whale fall. So a whale fall is when a whale falls to the ocean bottom, and what's remarkable is that there's like a whole ecosystem that's associated with that event. All kinds of things living there. These particular images are from the 2019 Nautilus expedition. These images are 3.2 kilometers down, and you can watch the film from which these are stills at that link there. But all kinds of vertebrates and invertebrates and just a whole unique ecosystem happens when whales fall. It's representative of the kind of diversity you see in the deep sea. It tends to be somewhat patchy, but as I say, highly diverse. But it doesn't actually have to be a whole whale that falls. All kinds of stuff falls down from the abundantly productive shallower waters and provides in the deep sea the nutrients that are necessary for life to flourish. And there's been quite a lot of discussion about why there should be so much diversity in the deep sea, all kinds of different hypotheses presented. But one thing is that there's just a lot of different types of niches in the deep sea, different habitat characteristics across the geographic range, different opportunities for life histories from scavenging to predation. And at the extreme, there's the necessity to adapt to extreme pressure, very low temperature, and the lack of light. So in general, in the deep sea, there's a lot of diversity, whatever the mechanisms. This talk is also about conservation. So I need to say a bit about what we do in the oceans. We dump chemicals which become bioaccumulated along food chains. We also introduce physical matter like plastics, which can be ingested or cause entanglement. And plastic pollution is not just about coastal areas. From this nice poster from the Scripps Research Institute, you can see these orange gyres called garbage patches in the Pacific. And those are actually drawn to scale. They're about 2 times the size of Texas. These massive patches of garbage. And about 70% of that falls into the deep sea. And of course, there are other places around the world where this kind of concentration can happen, including in the North Atlantic. This study was conducted close to one of those gyres in the North Atlantic. And they looked at 7 deep sea species, all of which had some level of plastic. And then amongst the 233 species that they looked fish they looked at altogether, 73% of them had plastics in. So it's about the kind of stuff we dump into the ocean, but also it's about what we take out. So as a species, we seem to be very fond of seafood. And this plot from ICES shows the progression of fisheries from 1950 to 2010. And the red line is for marine fisheries. And although it leveled off a bit in recent years, you can see that it's leveling off at about 80 million tons of fish per year, which is a lot of fish. And in this graphic from the Global Ocean Commission, they show that in 1950, only about 1% of the potential fishery species in the high seas was being exploited. And none of those were overexploited at that point. But by the time you get to 2006, 63% of the possible fish are being fished, and 87% of those are exploited, overexploited, or collapsed. And as the more accessible fish in shallower waters become depleted, the focus increasingly turned to the deep sea. And the techniques used to fish in the deep sea, dredging especially, often take a lot of bycatch, as you can see in the picture in the lower left. Starting in the 1970s, this fishery expanded considerably worldwide. And in the upper right there, you can see the various places around the world where there are deep sea fisheries. Well, historically, fisheries have been managed by species, and this is often still the case. For example, salmon fisheries, bluefin tuna, blue crab, etc. And a species' survival depends on its ability to respond to a changing environment, especially important these days with climate change. And the diversity required is often divided amongst populations within the species by region or by habitat. And these populations could be locally adapted or differentiated by random processes, so-called genetic drift, or both. And within these populations, the effects of pollution can affect growth rate, reproduction, and other aspects of fitness. I'm not going to focus on that anymore today, but it's worth bearing in mind. Okay, so what I'm going to do is I'm now going to focus on fish species in particular and species that we have studied on our lab. And I'm going to start by talking about geographic diversity or horizontal diversity, if you will. And the first example of fish is this guy, the orange roughy. Kind of a remarkable fish. They breed on seamounts, especially in the open sea, but sometimes also on coastal banks at around 700 to 1300 meters, although their full depth range is broader than that. It takes them 30 years to mature, and they live up to 150 years. So as you can imagine, once depleted, these guys are pretty slow to recover. Their eggs stay about 10 days in the water column. And this matters because currents move larvae and eggs around. And the longer they're in the water column, the further they can move. So this is a relatively short period. But they have moderate to high fecundity, about 25,000 oocytes per kilogram. On the left there is the plot of in the North Atlantic of where we collected samples, all the open circles there, both north and south of the subpolar front, which is a boundary where it's colder to the north and warmer to the south. So the orange roughy fishery peaked in the late 80s, early 1990s. It was a popular fish because its flesh didn't smell very fishy. But as you can see, over time, all over the world, the fisheries have essentially collapsed. So postdoc Tom White in my lab did a study on the effective population size of orange ruffy in the North Atlantic. So the effective population size, and distinct from the census population size, is an evolutionarily relevant population size. It's the size of the population that determines how diversity is lost over time. And by our calculations, which are to some extent approximate, but we got about 2,000 for the effective population size of the orange ruffy. But the catches were as high as 100,000 tons per year. And in that picture on the right, there's probably 2,000 fish in that net. So how can the number be so small? Well, as I mentioned, these are highly fecund animals, and if there's high variance in the family size among different fish, then that can depress effective population size. It's called sweepstakes breeding. And so that becomes important in terms of considering the conservation of these species, where they seem to be extraordinarily abundant, but the really important number, the effective population size, is actually quite small. So we then turn to looking across the whole genome for this species. It's a kind of genome sampling method we used, where we identified about 5,000 single nucleotide polymorphisms, so-called SNPs, and looked at about 1,000 fish. And this work was done by postdoc Enders, who was working at CSIRO in Tasmania with Phil England. And on the map there, the dots show all the different places where we got fish for this study. And this method is called an ordination method. When the dots are close to each other, it means the genotypes are similar, the genetics make them similar. And across the world, we see that the major regions are all separated genetically from each other. We just focus in on the Atlantic and neutral loci. So these are loci that behave neutrally over evolutionary time, as opposed to those that are apparently under natural selection. You can see there's some structure in the North Atlantic and the separation between the North and the South Atlantic. But then we can look at loci potentially under selection in particular by comparing the genetic distance among populations, in this case using this metric developed by Sewell Wright in the 1930s called FST, and compare that against the level of diversity within populations. And there is an expectation for what that relationship should look like under neutral theory, and that's shown in blue on the plot there. And the triangles in red, those are loci that are outside that range and suggesting positive selection. So if we look at the 295 loci that were outside that range and possibly under selection, what we find is that one population falls out, the population at the Faraday seamounts, and all the others are just together in a cluster. And if we just take 294 random neutral loci, then the Faraday samples just fall within that broader cluster with everybody else. Those are the bold Fs there. Well, that was pretty interesting, but it was using a relatively simple method to identify loci under selection. And so we worked at it again and used 4 different methods. And what we were especially keen to do is to identify neutral loci with good resolution. And so this time we got 3,700 neutral loci and about 420 loci that we could call outliers. And when we look at this again with the outliers potentially under selection on the left and the neutral loci on the right, and 2 different kinds of clustering methods above and below, we still get the same message. The Faraday seamounts fall out compared to everybody else as distinct. And looking at neutral loci, we find some pattern of structure in the North Atlantic. And so focusing first on the neutral pattern of diversity, one thing that often happens is that you get a pattern of isolation by geographic distance. So it makes sense because as populations are more distant from each other, then the rate of dispersal is probably less common. And if we look at that strictly amongst these populations, considering nothing but geographic distance, we do see something of a relationship, but it's not actually significant. The p-value is 0.06. But if instead we look only at geographic distance along the relevant depth range for this species, so 500 to 2,500 meters, then we get a much stronger correlation, and the p-value is much more significant. So the take-home messages from the Orange Ruffey study. There is isolation by distance, but it's along depth contours. And so in defining units of conservation, it's important to bear in mind the mechanisms that actually generate that pattern. And then there was apparently a differential local selection in one population amongst all of them, according to the analyses we did. And it's possible to identify genes that may be close to the loci that were outliers. And for the strongest outliers, we did look for gene functions, and they seem to be associated with sensory transduction and biological rhythms, although we need to do a lot more work before we're sure about that aspect. So in Halvor Knudsen's group in Norway, he's also looked at a number of other species, and here are a couple of examples where we see the same kind of pattern for neutral loci. So there's isolation by distance, but for tusk, it's along bathymetric barriers generating structure. And for the Greenland halibut, who has a relatively long larval period, and so the larvae are affected very much by current patterns. And so there's a lot of influence of so-called larval drift. And the population structure is influenced heavily by this pattern of currents. And for a couple of studies which are still in progress, for the short-beard grenadier and deep sea shark, we had samples from multiple populations, but including samples from around this Faraday area. So we just wanted to look to see if we see a similar pattern as we saw for the Ruffey. And what we see is using, again, this genome sequence sampling method. And for neutral loci on the left panels and outliers on the right panels, again, we have the samples from around Faraday falling out for the red arrow for the grenadier and the green arrow for the deep sea shark. For the deep sea shark, the red points are from a different ocean as an outgroup. And this is done by Michelle Gaither and master's student Luke Cartledge. Okay, so that was all about geographic structure, horizontal structure across the species range. And that's the kind of thing that people tend to focus on when they define stocks for management. Rather rarely do we instead think about population structure by depth, so sort of vertical in this system. But that's what I want to talk about for the rest of the talk. So as you go down in the sea, every 10 meters, pressure increases by 14.5 pounds per square inch of 1 bar. So at 1,800 meters, you're at 2,600 pounds per square inch. The ecosystem also changes because the potential prey and predator community changes, and then there's less light, and the water becomes increasingly cold. And there are potentially natural boundaries when you think about the structure of the ocean. So between the mesopelagic and the bathypelagic zones, you lose light. It's about a transition from about 1,000 to 1,500 meters. In middle latitudes, if you look in the upper right of the slide there, it also becomes more uniformly cold at depths below 1,000 meters. Salinity tapers off. That's the next graph down. And on the bottom graph, around 1,000 meters, you get the oxygen minimum and the nutrient maximum layers. So there's the potential for this being a natural kind of boundary. So the next fish I want to talk to you about is this round-nosed grenadier. It's living in a benthic habitat that is on the bottom and on slopes. It has quite a broad habitat range by depth, from about 180 meters all the way down to 2.2 kilometers deep. This species has a high potential for larval drift. This is again work from Tom White in my lab, and he genotyped 434 fish at 12 microsatellite DNA loci from all these sites that are shown in the map on the left in the black dots. From that study, we just found the one aspect of structure either side of the subpolar front, which is shown by a red dashed line in the map there. Amongst those 12 loci, there was one locus that appeared to be linked to some locus under selection. It also behaved as an outlier. So the population structure data is from the 11 presumably neutral loci, but we also looked at this one outlier locus and considered the allele frequency pattern according to depth. So in really deep water, the allele frequency pattern is quite different from what you saw in shallow water. And you can see that in those pie charts in the lower right. So this intrigued us, and we wanted to look harder at it. So Michelle, George, and Fatty, together with collaborators Neil Hall and colleagues at the Genome Center in Liverpool, we generated a reference genome for Coryphaenoides rupestris, including RNA sequencing, messenger RNA, so we could identify the genes, so-called annotation. And then we also resequenced an additional 60 fish at 5 to 10x depth. That is not as thoroughly as we had done for the reference genome, but enough to be able to do the analyses we wanted to do. This was also in collaboration with Francis Neat and co at Marine Scotland, who have this lovely boat they go out on, the Scotia. And they collected sample source from 750, 1,000, 1,500, and 1,800 meters. And we sequenced the genomes of those fish for 14 to 16 fish from each depth. And this is just a map showing where those samples exactly came from, all within about 20 kilometers of each other, and all collected at about the same time over a day or two. Okay, so again, here what we're looking at is a comparison using genome sampling, so lots of single nucleotide polymorphisms, lots of SNPs. And in the upper figures, that's again this sort of ordination plot where when the dots are close to each other, then the genotypes are very similar. And looking at neutral loci, there's really no distinction between the fish at these different depths. And really, the expectation might be that there wouldn't be, because they're all from the same place on essentially the same day, but just at different depths. But when you look at outlier loci, potentially under selection, there is differentiation according to depth. At the very bottom there, there's a table looking at that same metric of genetic distance among populations, FST. And for almost 45,000 of these neutral loci, there is no evidence of differentiation at all. So that suggests that whatever else is going on between these different depths, there doesn't seem to be a assortative mating, at least it's not showing up in these analyses. That is, deep water fish mating with each other and shallow water fish mating with each other. So then what we can do is we can look sort of along the genome and use depth as a trait and ask, is there any correlation between this trait, depth, and genotype anywhere across the genome? And that top red line is the most stringent of the significance values. And if we compare the shallow depths to each other's, for example, here 750 versus 1500 meters, really don't see much going on. But if we compare anything against 1800 meters, then it looks entirely different. And those stacks of dots are places where there's a strong signal for a correlation between genotype and depth. And we can take all of those outliers, all of those positions within the genome that showed a significant deviation, and identify genes nearby, and we get a list of genes. And we can ask if that list of genes shows gene functions that are significantly distinct from the overall background set of genes for this species. And the answer to that question was yes, for this set of gene functions here. And most of them had to do with morphogenesis or development. So something to do with shape and phenotype. If we pick out 9 of these outlier SNPs, shown in the right there, and compare the different depths, what we see is that at 1800 meters, they have fixed difference compared to everybody else. And these 9 loci are within 6 genes, which are listed at the bottom of that paragraph on the left. And there are 1 to 4 non-synonymous changes within these loci. So all of these changes highlighted here are non-synonymous. That means that when you change that part of the DNA sequence, it also changes the amino acid sequence. So it changes the protein. So it potentially changes the function of that protein. If it was synonymous, then there would be a nucleic acid change that did not actually change the amino acid. Now in this figure, we're looking at the relationship between physical distance along the genome on the x-axis there, and something called linkage disequilibrium on the y-axis. So in linkage equilibrium, there would be no association amongst loci. But in linkage disequilibrium, there's a non-random association amongst alleles at different loci. So the expectation here for neutral loci is that when closer on the genome, they would show higher linkage disequilibrium than when distant. And that's just because there's less recombination going on between the sites when they're close on the genome, when the physical distance is close. But other things can actually elevate linkage disequilibrium, implying some kind of association between the genes, and that can include selection. And so for our outliers in blue, that's just all the outliers that we found. And then the ones in red are the non-synonymous changes. Many of those show a higher level of linkage disequilibrium than you'd expect for distance separation along the genome. So that suggests that they're pitching together, as it were, on a similar kind of function. So then we took 4 of these boxed there, particularly strong ones, and compared them across the species range in the North Atlantic. All these different sites, all the different blue dots there, and asked what proportion of the individuals had the so-called deep allele. And when we look at juveniles, which tend to be found at 1,000 meters, there's no strong relationship there. They're just about 50-50 for alleles, deep water allele versus the not deep one. But when we look at depth, no matter where it was in the North Atlantic, for adults, the deepest water fish had the highest proportion of deep water alleles, and the shallow water fish had the least. So the point here is that it's a phenomenon that's not just about where we sampled and studied and did all those genomes. It's happening across the species range. And this plot shows where you find fish of different size, and size correlates strongly to age in this species. So our youngest fish, the shortest fish, are typically found at depths of 1,000 meters or lower, but the adults can be found either around 1,000 meters or deeper. And collaborator Thomas Regnier at Marine Scotland did this lovely latent class model here based on tremendous database of catch per unit effort data. And here's just illustrating more clearly the fact that there are two different kinds of strategists as adults. So on the left there, you see that the young fish are at about 1,000 meters, essentially always, and then the older fish either go to deeper water or they stay at about 1,000 meters. And this is just to illustrate again that for the juvenile fish, the allele frequencies are about even, but for adults in shallow water, it's biased one way, and for adults in deep water, it's biased the other way. And here I want to focus in on just one of these loci because it's a really interesting one. This is obscurin, and master student Tash Steeds did this work. It's a very long protein, and it's illustrated in pale blue on the bottom there, also marked C in the figure. And it interacts with an even longer protein called titin. And basically it's a structural molecule within muscle fibers. And on the top is some modeling that Tash did. And the red bits are showing the places where there's a difference between the shallower water and the deep water fish. So those are the places where those mutations occur. And what we found was that for the deep water versions, those alleles lose the potential for intra-domain bonding. The same kind of pattern is seen all the way across the length of these long proteins. What it suggests is increased flexibility, but decreased stability, and a reduced ability to reform correct shape if the molecule is overstretched, and that's for the deep water version. What that means actually in terms of function for the fish, we don't really want to speculate. We haven't done any experimental work, but the important message is that there are changes that are potentially functional associated with these deep versus shallow water mutations. Tash also looked at morphology. She found that in the deep water form, they had a bigger gape size of their mouth. On the lower left there, they had increased liver mass. They had more elongated bodies, so long and thin compared to shorter and fatter. And if we take all these aspects together, and build a so-called random forest model, we can use that algorithm to identify fish based on their morphology as to where they most likely came from with rather small chance of error. And visiting PhD student Zulkarnain has started to look at this at 3 more species of fish, focusing on the morphology. And the lower left fish there, it has a similar kind of mouth shape to the grenadier, and it shows the same kind of pattern with greater gape size at depth. The middle fish has a very different kind of mouth phenotype, and it shows the opposite pattern. And then on the right is the deep water eel, which has an absolutely immense gape, and it shows no pattern at all. So the main message here, though, is that it's not just for the grenadier. For other species, there are also morphological differences from the same part of the sea for the same kind of comparison of depth, either side of that putative boundary around 1,000 to 1,500 meters. And then a PhD student, Daniel, took some time off his work with dolphins to look at the grenadier stable isotope patterns, and this tells us something about feeding behavior. And he found that there was a significant difference between the deep water feeders and the shallow water feeders. Okay, so the take-home messages from the grenadier study. The young fish remain mostly at 1,000 meters. We don't actually know precisely where these fish breed, but there are swarms seasonally at around 1,000 meters, and it's speculated that maybe that's what's happening there. Then as adults, they segregate by depth, and they do so apparently by genotype, which is pretty interesting, but also by morphology. This is not unique amongst fish species. There are many species where the ecotypes in different habitats, even within small lakes, can have very different forms. Some aspects of this may be plastic. It might develop during development in different habitats. But in this case, there are clearly strong genetic differences between these types. We found no evidence for assortative mating, and so no evidence for incipient speciation. And in addition to the genotypic differences, there's phenotypic differences and also differences with respect to diet at the different depths. Okay, well, what I want to do now is just finish with another boundary, and this one between the bathypelagic and the abyss. And it's typically taken to be about 4,000 meters. And at that depth, the pressure is extremely high. There's a constant, very low temperature just above freezing, and there's no light at all. And so in this study, looking at 2 mitochondrial and 2 nuclear loci and building a phylogeny, we see that the abyssal species amongst a number of species of Coryphaenoides all follow in the same lineage. And then with Michelle and George and Fatih, we again looked at genomic data. In this case, we're looking at mitogenomes, so the mitochondrial genomes. And this tree shows a clear separation between 7 deep water species that we focused on the top there, and then in the circle, 7 abyssal species. And we can use methods to date nodes within the tree. And when we do that, we get a separation between these lineages of about 21 million years ago. So the Paleogene was a period when the ocean basins were being formed. It's a relatively long period, and it finished about 23 million years ago. So it's possible that this radiation happened in association with that. We then looked at the nuclear genome, and we identified non-synonymous changes within genes that were fixed different in our deep water species compared to the abyssal species. And there were very, very many of these. This is just a small sample. But notice that amongst them are obscurin and titin, shown in the blue arrows there, where there's a fixed non-synonymous change. In total, there were 12 loci associated with muscle function and another 65 associated with membranes. And the most significant functions identified from this list when we do that kind of gene function analysis were for transmembrane proteins and signaling receptor activity. So another thing we can look at is, oops, sorry. Oh dear. So another thing we can look at is this relationship between non-synonymous and synonymous change within a phylogeny, and in that way identify loci that are potentially under selection. And when we do that, we get a number of significant loci coming out of various functions, but including in green, a number that are associated with membranes, and in pink, the number that are associated with muscle function. And then another thing we can look at is a copy number of genes. So sometimes genes are present in different numbers within the genome. And this list of genes here showed variation in copy number. And the color pattern is showing you fold difference. That's how many times different it is. And the red bar at the top there is identifying the abyssal species. And orange means larger copy number. So for all these loci, there's greater copy number. And when you look at those, the functions for those loci, very many of them have to do with membranes. So just to be clear, there's lots of functions identified by these analyses we need to look at harder, some of them having to do with protein folding and various other things. But there was an awful lot of signal for membranes. So it raises the obvious question, why membranes? Well, there's this study by Kato et al., which took a pig membrane and subjected it to different pressures, including the kind of hydrostatic pressure you might find in the abyss. And they found a decrease in the fluidity of the lipid bilayer in the membrane. They found a conformational change in the transmembrane proteins, and they found functional disorder of membrane-associated ATPase activity. In other words, it messed it up. And if they put more pressure on, it kind of fell apart altogether. For marine fish, there have been a number of studies, and in general, they find that hydrostatic pressure is associated with disruption of signal transduction. And then there was actually a study by Seibenaler in 2000 where they looked at 4 Coryphaenoides species of the same genus we were looking at, and including 3 of the same species. And he looked at adenylyl cyclase activity, which is a transmembrane protein important for intracellular signaling pathways. And he compared species that had different depth ranges. And as you can see at the bottom there, on the left, there's a relatively shallow water species. And although for both species, you get a diminution of activity for the enzyme with depth, it's less so for the deep water species. And this is a locus at which we also found one of these fixed non-synonymous changes. Okay, so the general take-home messages from the abyss work include that there's really quite extensive difference between this group of species living in the abyss compared to a group same size group of species living in simply deep water. So there's clearly something going on either side of that boundary. The relevant functions are about a lot of different things, but membranes and muscle structure and function seem to be particularly important. And although the overall emphasis was different for the boundary at around 1,000 meters compared to 4,000 meters, there was some shared loci. However, mostly at 1,000 meters, it seemed to be about development and morphogenesis, whereas in the abyss, it was really about stuff that was probably being affected by very high pressure. So what kind of conclusions can we take from all this with respect to conservation? Well, one thing is that typically when managers divide populations into stocks, they consider geographic populations, and sometimes they're just largely about sort of political divides even. But what we really need to understand is that there are environmental drivers for population structure that affect the way populations are structured in the sea. And sometimes these are not particularly obvious. And so we need to consider those structures when we identify populations for conservation. And it's also possible for populations to show essentially no differentiation at neutral genetic markers, but strong signal for local adaptation. And so in order to conserve adaptive potential, we need to be able to identify those distinctions. When we shift to considering populations by depth, what we see is that for the example I talked about, there's little evidence for differentiation at neutral loci, but very strong evidence for differentiation by selection. And what seems rather remarkable to me is that you have animals that are probably breeding and growing up to a certain extent at a particular depth, around 1,000 meters, and then segregating according to genotype, and perhaps by phenotype, to one depth or another, only as adults. And so if you imagine that you have a fishery for a particular species at a particular depth, it's possible that you would deplete the genotypes required to live at that depth unknowingly, thinking that, well, they're all the same fish at that particular location. So again, something probably we need to pay attention to. And with that, I just want to thank our funders for this work, and again, all the various people who have contributed to it. And I thank you for your attention.